Hello folks, I'm Nikhil Maland, and today we are going to be talking about immunohistochemistry. I'll be doing a study regarding the application of immunology in pathology, specifically facilitated by the procedure of immunohistochemistry. Before we start jumping into a complicated topic, let us ask ourselves, what is immunohistochemistry? Well, as you may have already noticed, it is a word that is composed of three distinct fields of sciences. The first of these, which comes from immuno, is immunology. Immunology is the study of the immune system that functions to protect organisms from foreign pathogens. The second term, which comes from histo, is histology. Histology is the study of tissue preservation, and tissue staining. It is a tool used widely for studying the pathophysiology of organisms. The final term is chemistry, which is the study of matter. In this context, we utilize our understanding of chemistry to help histology. Thus, we can now create a concrete definition of immunohistochemistry, or IHC. IHC is a set of procedures used to stain tissue samples using the chemical interactions that occur between the immune system and the molecules inside of the tissue. Now, let us try to understand why IHC is necessary. So why do we stain tissue in the first place? Let us analyze this tissue sample that was stained. The staining procedure used on this tissue sample is actually an extremely common stain called the hemotoxylin and eosin stain. The staining is so standard that it is even referred to as the golden standard of histology. Looking closely at the slide, we can actually tell why staining is so essential. By staining the tissue, we can see the different structures that are present. We see the top layer of keratinized fibers, followed by a densely packed layer of keratin. Following this layer is epithelial tissue, under which we see adipose tissue. We also see other glandular organs. This is a sample of skin tissue stained with standard hemotoxylin and eosin procedures. We can appreciate why histology is so integral to pathophysiology. It is almost impossible to identify tissue and structures without a proper stain. Hemotoxylin is the first chemical used in the stain. Hemotoxylin is responsible for staining the nucleus of all cells blue. Hemotoxylin by itself does not produce any staining. Rather, treatment with other chemicals and ions leads to the final stain. The tissue is then counterstained with eosin. This counterstain stains proteins and other acidic or negatively charged ions pink. Together, the eosin contrasts the hemotoxylin. Furthermore, different cells stain different amounts when eosin is introduced, allowing for structures to be highlighted in the tissue. Well, by the looks of it, histology sounds like a glorious field. Pathologists most definitely utilize histology as a method of tissue analysis. However, there are important issues that histology cannot overcome with simple tissue handling and staining. When specificity becomes more valuable than simply viewing structures, we rely on immunohistochemistry. One example of when immunohistochemistry is important is when we want to determine the type of cell in the tissue sample. Many cells can look alike and are not necessarily differentiable simply through analysis of structures they belong to. One such example is when we analyze tissues with different types of cells that have similar morphologies. Here's an H&E stained slide of a uterine cancer. The predominant morphology displayed here is that of spindle-shaped cells. The population of spindle-shaped cells can be of many different types, since cancers tend to make cells pleomorphic, which means that they become deformed. In this case, the cancer cells may originate from endometrial stromal cells, smooth muscle cells, Schwann cells, or even macrophages. Here's another stain, this one accomplished using IHC. Here, we tested specifically for a protein known as SMA, or smooth muscle actin. The cool thing about SMA is that it is only found in smooth muscle cells. Since most of the spindle cell population is staining for SMA, we know that the cancer cells originate from the smooth muscle, making this a leiomyosarcoma. So IHC is really important for finding extremely specific things in tissue samples. IHC also provides interesting ways of visualizing tissue structures inside the cell. This is demonstrated with these stunning images. In these cells, IHC was used to specifically highlight the cytoskeleton and mitochondria inside the cell. Since most common histological stains can only highlight polycellular structures, we see why IHC is of such importance to histology. It is primarily an extremely specific procedure for isolating a certain population of cells or looking for the presence of a certain type of molecule in the cell. Furthermore, it revolutionizes the visualization of tissues and cells by providing an in-depth visual of cells and cellular processes. IHC can get extremely complicated if I delve too much into the topic too quickly. Rather, I'll give a general idea of all the procedures and then narrow down into most of them. Remember how H&E stains use a hemotoxylin stain and an eosine counterstain to stain the tissue? Well, these two chemicals are called staining reagents. The goal of a staining reagent is to be able to locate certain molecules and attach them inside the tissue that has generally been treated in a certain way. Furthermore, the staining reagent must then be able to change the color of itself or the objects locally around it so as to make the identification visible. In immunohistochemistry, we utilize antibodies as our staining reagents instead of standard and broad-acting stains used in histochemistry. When tissue is extracted from an organism, it requires preservation. In histology, the standard technique of preservation is to embed the tissue in paraffin wax and fix it with formalin. After removing the blood, tissue is placed in a formalin solution for fixation. Fixation holds the structure inside the tissue in place. Tissue that is obtained from patients and organisms is often embedded in a wax block of paraffin. All tissue needs to be embedded before it is cut for slides. Here, we have a picture of certain tissue samples that have been embedded. After fixation, the paraffin blocks are cut into thin sheets using a microtome. The microtome is a tool used by histologists to put tissue slices onto slides so that they can be viewed under the microscope. Unfortunately, formalin fixation and paraffin embedding of tissues leads to several problems. Antigens cannot easily find molecules inside the tissue once the paraffin is removed, since formalin fixation is relatively permanent. To combat this issue, a process of antigen retrieval is conducted. AR, or antigen retrieval, involves using enzymes, heat, or a multitude of other methods to make it easier for antibodies to find antigens. From here, a diverse set of methods can be applied to perform IHC. The first method used by researchers was known as direct labeling. Direct labeling involves an antibody finding its desired molecule. On this antibody is a certain type of label that allows researchers to see the antibody. Thus, when the molecule is found in the tissue, it stains visibly. Similar to this method is the indirect method, which uses two antibodies. The first antibody, known as the primary antibody, attaches to the target molecule. The secondary antibody then attaches to the primary antibody. Finally, a label, which is generally an enzyme, on the secondary antibody allows the researchers to see the stain. Another common method used in IHC is known as the avidin-biotin complex method, or the ABC method. When researchers realized that the indirect method did not provide enough staining, they attached a lattice full of labels to the secondary antibody to make sure that the stain was visible. A molecule called biotin was attached to the secondary antibody to anchor the lattice. The lattice itself is made up of avidin molecules with 4 biotin molecules surrounding it. The biotin molecules attach to label molecules, which in turn attach to more biotin molecules. Another common method is polymer-based IHC. Generally, a labeling complex is made before the procedure is even performed. This complex has a dextran polymer as the backbone for the labeling complex. The polymer has secondary antibodies attached to it. These antibodies will target the primary antibodies that have already located the target molecule. Furthermore, labels are attached to all free moieties on the polymer. Generally, this polymer has up to 70 labels. This complex then attaches to the primary antibody and allows for the stain to be visualized. Certain issues related with polymer-based immunohistochemistry led to the development of catalyzed signal amplification, or CSA. In CSA, we begin by completing the normal avidin-biotin procedure that was discussed previously. Another complex is used to add staining close to the target molecule. Another protein like avidin, known as streptavidin, is used to make this complex. Labels are added to the streptavidin to make the stain more visible. Finally, a biotin molecule attached to tyramine is attached to the streptavidin. When the streptavidin complex is added, the tyramine reacts with labels previously attached through ABC and becomes sticky. This sticky complex sticks to molecules close by the antigen, thus providing further staining. Optionally, one can also use fluorescent tyramine instead of streptavidin and biotin to view the stain with fluorescent microscopy. Okay, so now that we have a general idea of what's going on in IHC, let's get to the specifics. The first part of IHC is getting the correct antibodies. Antibodies come in two forms, monoclonal and polyclonal. Let us take our discussion to polyclonal antibodies first, because they are similar to the antibodies we have in classical immunology. The first thing to realize is that we cannot actually use human antibodies for immunohistochemistry. Why? Well, think about what actually makes antibodies in our body. Antibodies are made by B lymphocytes. B lymphocytes, after maturation, have a unique receptor that matches an antigen. A professional antigen-presenting cell usually presents the antigen to a helper T cell that then activates the correct B lymphocyte. The details of how T cell to B cell activation occur are not very well understood. However, from our last adaptive immune system video, we know that T cells go through maturation and testing in the thymus. During testing, we make sure T cells don't respond positively to human cells. Therefore, we know that human B cells will never respond to proteins that are human. We therefore cannot find proteins using IHC that are human in origin. It is therefore common practice to use other animals as a source of antibodies, such as mice, horses, donkeys, goats, and rabbits. Polyclonal antibodies are made by presenting the antigen to these animals. Once the animal gets exposed, its own body begins producing serum containing multiple antibodies. These antibodies are created by the B cells in the animal and attached to antigen that was presented. Every antigen can have multiple epitopes to which different types of antibodies can attach to. Therefore, it is not surprising that multiple antibody flavors are found in the serum. Due to the use of antibodies that target multiple epitopes, these antibodies are called polyclonal. Polyclonal antibodies target multiple epitopes on the antigen that was presented to the organism. However, cultivating antibodies in an organism is an arduous task. Researchers eventually began looking toward a method that could simply generate antibodies of a certain type. This occurred for multiple reasons. Firstly, in IHC, we require reliability of testing procedures. That is, every time the antibodies are used, their effect should be determinable. Polyclonal antibodies go through multiple stages of purification before being used in histology. However, since every organism may produce a different mixture of serum that targets different epitopes, the effect of the antibody is often unreliable. For example, the serum during general testing may work very well. However, another batch of the same antibody may have antibodies that have an affinity for proteins other than the target antigen. For this reason, researchers looked for a method of isolating one antibody once it has been confirmed as a useful antibody and generating clonal copies of it. To create such monoclonal antibodies, researchers most often use the hybridoma technique. After presenting the antigen to the organism, we remove the B cells from the spleen that were producing the antibody. These B cells are then fused with myeloma cells. A myeloma is a form of cancer and is a well-categorized immortal cell line. Specifically, myelomas with the HGPRT deficiency. There are two ways by which cells are biosynthesized nucleotides, through the de novo pathway and the salvage pathway. Without HGPRT, the salvage pathway stops working. Thus, the myeloma barely functions. When fused with a B cell, it forms a hybridoma. Since the B cell has HGPRT, the hybridoma can access both pathways once again. The hybridomas are grown in a hypoxanthine, aminopterin, and thymidine medium. which kills cells without the ability to use the scavenger pathway. Thus, only hybridomas are left in the cell culture. Once the hybridoma has been created, it can produce monoclonal antibodies for as long as you want it to. So when do we use polyclonal antibodies? And when do we use monoclonal antibodies? Well, we know that polyclonal antibodies attach to multiple epitopes of an antigen. For example, if we were targeting a protein called smooth muscle actin, which is a rather large protein, the polyclonal antibody serum would have antibodies that attach to many different locations, or epitopes, on the protein. However, monoclonal antibodies would consistently attach to one part of the protein. Monoclonal antibodies are useful for finding proteins with consistency over a long period of time. This allows researchers to study one protein over multiple batches of the serum. In contrast, polyclonal antibodies can vary. However, polyclonal antibodies are more likely to actually show the stain because more antibodies attach to the protein. Therefore, the use of monoclonal or polyclonal antibodies is determined on a case-by-case basis, depending on the protein, the tissue sample, the conditions, and the number of epitopes involved. For example, monoclonal antibodies have a small range of pH that they are functional in because all the reagent molecules are the same. However, in polyclonal serums, the multitude of antibodies allows for more staining in a larger pH range. It is of value for us to now define two terms that are commonly used to describe staining in tissue. Specificity is the percent antibodies that correctly bind to the target molecule and stain the correct cells. An antibody that has a higher specificity is therefore more accurate. The opposite of this is non-specificity, which describes staining that was consequential or unexpected. Here, we see two samples of mouse pancreatic tissue. The sample on the left does highlight the islets that are found in the pancreas. However, we also see a lot of staining that seems to be nonspecific. The sample on the right, on the other hand, correctly identifies islet cells and has minimal nonspecific staining on other cells. Other than these two terms, we also think about IHC procedures in terms of sensitivity. The sensitivity of an IHC procedure can loosely be defined as the percent of target molecules that were revealed through staining. Thus, a more sensitive antibody has the ability to find more proteins, or consequently, give a stronger stain for proteins that are found in low concentrations. Now we have covered two specific portions of pre-IHC protocol, producing the antibodies and choosing the right antibodies for the job. We'll now start talking about a generalized IHC procedure. Let us begin with a discussion of formalin fixation and paraffin embedding. Remember that we embed tissue samples in paraffin so that we can properly cut the tissue. Think about it this way. If you try to cut anything that is kind of squishy, the cut is usually uneven, diagonal, and squishes whatever you're cutting. When talking about tissue samples, this means that some regions have a higher number of layers of cells than other regions in a cut piece of tissue. To keep the tissue sturdy whilst being cut, we embed it in paraffin to make it into a relatively solid block. We then use a microtome to cut blocks with precisely measured thickness and place them on slides. Once the paraffin block is placed on a slide, it is archived until immunohistochemical analysis is needed. Why do we fix the tissue in formalin before making a paraffin block? Well, let us try to understand why we need fixation in the first place. Cells are compartmentalized heavily. However, when the organism dies, autolytic enzymes in the cell begin destroying the cell from the inside out. Furthermore, fixation also makes the membranes of cells more porous. This allows for antibodies and paraffin to actually penetrate the cell. If antibodies are unable to get into the cell, IHC procedures would not be possible. Thus, fixation is a bare necessity for IHC procedures to occur. However, fixation works by a process called cross-linking. Formaldehyde is an extremely reactive molecule that forms polymers in aqueous solutions. These polymers form bridges between amino acids and proteins. Thus, proteins stop functioning but retain their structure even after dehydration. Since a huge part of protein folding is hydrophobic interactions due to an aqueous environment, dehydration of the cell can completely change the folding structure of a protein. Formaldehyde cross-linking bridges prevent the structure from moving. However, formaldehyde also has the effect of masking the epitopes on proteins. Although not studied extensively, the effect of masking due to fixation is well known in labs. IHC procedures could not develop successfully for many years because fixation, which we know is crucial for IHC to work, is itself masking and hiding the target proteins by covering up epitopes. Since fixation and embedding is required for histological and immunohistochemical procedures, what do we do if the epitopes can't be found by antibodies? Well, breakthroughs in a process known as antigen retrieval, or AR, have allowed IHC procedures to develop over the past few decades. AR procedures are basically used to unmask antigens that have been masked by fixation and other factors. Before AR procedures were effective, proteolytic enzymes were used to unmask epitopes in tissues. However, proteolytic enzymes are naturally degrading and may lead to the loss of structure in the tissue samples. Eventually, a link was found between heating proteins and the removal of formalin bridges in the protein created by cross-linking. Thus, a new age of AR procedures dawned in IHC history, that of heat-induced epitope retrieval, or HIER. Heat-induced epitope retrieval involved heating up the tissue sample after it had been fixed and embedded. This actually causes most of the formalin bridges to break down and unmask epitopes with great efficiency. After rehydrating the tissue sample, HIER is performed to unmask epitopes so that IHC procedures can be utilized. A proper pH buffer is used to heat the tissue, which is optimally around 8 to 9 in range. Researchers initially started using the direct method. However, there were multiple issues with this form of staining. The direct method's specificity greatly depended on the serum that was being used. However, it lacked sensitivity. With just one antibody attached to the target molecule, there was only one label that could be attached per protein. The label molecule that was most commonly conjugated with antibodies was the horseradish peroxidase. After adding colorless substrate, the horseradish peroxidase, or HRP, converts it into a stain that can be viewed through the microscope. However, stains created using the direct method are extremely light and hard to see. This led to the creation of the indirect method. Since more than one secondary antibody could attach to the primary antibody, there were multiple labels present at the target molecule site. However, even this method lacked the sensitivity that would allow for pathological and histological use of IHC. The ABC method revolutionized the sensitivity of IHC methods. The basic idea was to attach a lattice of HRP molecules to stain the target molecules with increased sensitivity. The ABC method was the first method that made IHC staining a viable option for histological and pathological analysis of tissue samples. The lattice was created using the interactions between two molecules, avidin and biotin. Avidin is a molecule found in egg whites and is readily available. Avidin has 4 binding sites for the vitamin biotin. Biotin can be conjugated with label molecules, such as HRP. Therefore, the HRP conjugated biotin attaches to the avidin in 4 locations. The HRP molecules then cross-link with other biotin-avidin complexes to create a lattice. The secondary antibody used to locate the primary antibody is also biotinylated, so that the lattice can attach itself to the secondary antibody. Thus, the ABC method increased sensitivity by adding a humongous amount of label molecules to the procedure. Eventually, newer methods of finding the target molecule began taking hold. The polymer-based immunohistochemistry procedure has a pre-made complex with the secondary antibody. This reduces the number of steps required to attach a lattice of label molecules to the target molecule. Here, we can see how each procedure is compared on a relative scale of sensitivity to number of steps. We are of course looking for a procedure that minimizes the number of steps and maximizes the sensitivity. The direct method requires the fewest number of steps, since the label molecule is attached to the primary antibody and simply needs to be put into the tissue. However, it lacks sensitivity and is generally not useful for IHC. The indirect method does provide some level of sensitivity and requires fewer steps. However, it is evident that integration of ABC methodology is required to boost sensitivity in the indirect method. The ABC method, therefore, requires 3 steps, whilst the indirect method only requires 2. The most optimal IHC procedure is the polymer-based IHC. The first step is to attach the primary antibody. The second is to apply the polymer complex, which is already conjugated with label molecules and secondary antibodies. Thus, polymer chemistry is the most effective IHC procedure. CSA2, while extremely sensitive, requires up to 4 steps and consumes a lot of time. Therefore, CSA2 is reserved for proteins that are hard to detect. Overall cost analysis, however, would suggest that the ABC method is also a viable option compared to more costly polymer IHC. Two diagnostic labels are commonly used in IHC, enzymatic and fluorescing. HRP is an example of an enzymatic label. It turns colorless substrate into a stain, thus elucidating the location of proteins in the tissue. Fluorescent tags do not require the addition of substrate. Rather, once the label has been attached to the target molecule, a peak excitation wavelength light source is used on the tissue sample. Electrons in the label jump to a high energy state. The extremely high energy state is quickly reduced to a semi-stable state due to excitation decay. As the electron finally returns to its ground state, it releases a photon of a different wavelength than that of the excitation wavelength. Commonly used fluorescent tags include fluorescein isothiocyanate and tetramethylrhodamine isothiocyanate. IHC staining methods provide immense histological insight into tissue samples and their pathophysiology. Rather than simply seeing structures, IHC allows us to illustrate tissue samples using various methods to not only view the tissue, but create an intricate story around its past. If not for research, IHC simply provides us a snapshot of cellular biology and the intricacy of life itself. But where does our understanding of immunology intersect with the field of IHC? Well, there is no one spot in the process we can pinpoint, since our comprehension of the immune system permeates throughout every IHC method we use. Our understanding of antibodies is vital for IHC. We have learned that monoclonal and polyclonal serums are important in immunohistochemistry. The electrostatic interactions between the antibodies and their FAB region and the epitope on the antigen results in specificity that enables IHC in the first place. Furthermore, the FC region, which is comprised of the heavy chain of the antibody, is a protein that can be targeted by secondary antibodies. The fact that antibodies are specific to a given epitope is the result of the mechanism of the immune system itself. VDJ recombination of the heavy chain and VJ recombination of the light chain provide for diversity in the immunoglobulin epitope interactions. Thus, helper T cells that can recognize virtually any epitope are produced by the immune system. These helper T cells are then capable of activating B cells that will become plasma cells. These plasma cells produce the serum that is used in IHC. When producing polyclonal antibodies, animals have professional antigen-presenting cells that recognize the target molecules as a pathogen and present it to helper T cells to begin the production of antibodies. The production of monoclonal antibodies takes this one step further by creating an immortal plasma cell line. Our understanding of continuous and discontinuous epitopes allows us to theorize why cross-linking proves to be an issue for IHC during fixation. Furthermore, it also allows us to understand why unmasking epitopes is so crucial to IHC. Thank you for watching my presentation on immunohistochemistry. I hope it was as enjoyable as it was for me to learn about this subject.